discussion, and I invite all of you to ask questions to all of the speakers. So please, we have a second line, was the first one. The microphone, please.
Hi, Neil Osterwile with Oncology Practice. A question for perhaps any of the panelists. So what do you think might be the mechanism, maybe I'll start with Dr. Schmidt, what might be the mechanism of action? Is it synergistic? Is it separate mechanisms? Why do you see such a good benefit with adding the immunotherapy?
That's an excellent question. I think if you look across different cancers, we see there's a clear interaction between chemotherapy and immune therapy. So one thing is chemotherapy releases antigens because chemotherapy destroys cancer cells. That leads to the release and shedding of antigens, which the immune system then can detect. Chemotherapy also disrupts the structure of the cancer, makes it easier for immune cells to get in. Chemotherapy, so we know that immune cells, CD8 positive cells, increase after we start chemotherapy. So on many levels, chemotherapy can facilitate for the immune system to recognize cancer. If you then boost the immune response by giving immune therapy, we have a synergistic activity, and that's what we see across multiple tumor types, including breast cancer.
Professor Sleiman, can you comment on the data?
I think it's a great question, and I think the answer is a little bit different in the sense that the two different therapies hit different pathways, both of which are important in the hormone receptor positive, HER2 negative subtype. So it's not so much that it reverses resistance to endocrine therapy, but it adds another pathway to be attacked. For the last 4.5 decades, we've spent most of our time working on hitting the estrogen receptor pathway. And I think with the mTOR inhibitors and now with the CDK4/6 inhibitors, we're seeing that this approach makes an impact.
Professor Sledge, any comment?
I agree with Dr. Sleiman.
Okay. We move to the next question from Fine.
Yes, dropper to BSB for Professor Schmidt. Given the heterogeneity we know exists in triple-negative breast cancer, have you yet obtained any insights into who responded and who didn't? Were we able to look at some molecular subtypes, for example, or histological subtypes, or stromal TILs to gain any insights as to who might benefit most from the addition of pembro, to chemotherapy when we are getting quite high PCR rates, as you said, with chemo plus platinum, and I guess I'm thinking in terms of the idea of, will we be able to select for the patients who need the pembro and the cost and toxicities associated with an additional line of therapy, or alternatively, would you be idea being that you would, if available, would everybody receive this optimally?
So these are 3 questions. So.
Yeah, no, thank you, Peter. Excellent question, as always. I mean, we have a lot of tissue in these samples and in these neoadjuvant studies, and I think in contrast to metastatic breast cancer, the tissue is taken right before we start the treatment, and then we have tissue at the end of treatment, at least in patients who didn't respond as well as we hoped to. So we will do all the analysis you suggest, looking at different subtypes, looking at molecular markers. But the other thing to mention is PathCR is one endpoint, but EFS is a different endpoint. And I personally expect to see a possible benefit of immune therapy even in patients who don't achieve a PathCR. So we will have to wait for some of those correlative studies quite long until we know we actually have prevented recurrences in these patients. One of the things we know is the tumor biology is very dynamic. And when we start giving chemotherapy, a PD-L1 negative tumor may turn into PD-L1 positive tumors within a few weeks. And I think this dynamic process, this plasticity of tumors is something we will take into consideration when we analyze the data better. The purpose is very clear. We need to identify the patients who benefit most, and patients who do not benefit as much. We need to see whether we can get away with less treatment or come up with better treatments.
I would like to have another point of view. Professor Sledge, what do you think about this?
In terms of?
In terms of identifying subpopulation of patients that will better respond to immunotherapy.
I think these are drugs that have real toxicities and are quite expensive. So to the extent that we can identify subpopulations that gain benefit, we'll be doing a good both for our patients and for society.
Okay, another question? Yes.
Hi, Caroline Helwick on the ASCO Post. The control arm looked really pretty good at 40 months or so overall survival time, and Dr. Harbeck told us that typically registries show 2.5 years. So is fulvestrant performing especially well as an endocrine agent, maybe better than some of our more commonly used, alone as a single agent?
So I think first thing to say is that patients who go into trials are not the same patients that you see on registries. They tend to be younger, they tend to be healthier, and they tend to be treated in centers where they have a greater likelihood of getting further therapy after treatment. So I think anything that compares a registry data to a trial data is probably inherently flawed because of that.
Okay. And for Dr. Sledge also, were your patients second line only or second line and beyond second line heavily pretreated?
So patients could only have had one prior endocrine therapy.
Okay. Thank you.
Mm-hmm.
So any other question? Yes, there.
Alex Castellino from Medscape. This is a question relevant to the CDK4/6 inhibitors. What do we know about them? We have 3 of them now in the market. What do we know about the resistance mechanisms across them? Are they common? Are they overlapping? Can one be used following another? How would you sequence them in practice, or do you?
I think people have been using one after another, but our own preclinical data, and I think some of the early clinical data are, with the exception of some, anecdotal issues, for the most part, there's cross-resistance. If you are resistant to one, you're resistant to others in the class. The things that are resistance mechanisms de novo is if you have a non-functional retinoblastoma protein, that certainly doesn't make the drug that allows for activation of that very, very effective. If there's amplification of cyclin E, in other words, getting past that checkpoint and entering the cell cycle by another cyclin CDK partnership, that's another way that there's resistance. So there are resistance mechanisms that we're beginning to follow up on, but clearly I think there's more to be learned in terms of the biomarkers. As George said, it's always important to identify the populations that are likely to benefit as well as those not, so that we avoid using drugs where they're not likely to be effective and use them where they're likely to be most effective.
So we have another question front line, and then, yes.
Yes, thank you. It's Jacob Pleath from Vantage in London. Dr. Schmidt, a fairly basic question, forgive me. Does this study support the use of Keytruda as neoadjuvant or as adjuvant, or both?
That's an excellent question. Ultimately, we cannot separate how each of the phases contributed to the overall benefit. At this point in time, we're looking at path CR rates, and that's obviously determined by the neoadjuvant part. But when we look at the recurrence of event-free survival, that's driven possibly by the neoadjuvant and the adjuvant part. So ultimately, the regimen that was evaluated was neoadjuvant chemotherapy plus pembrolizumab, followed by adjuvant pembrolizumab. What we can't answer in this study is whether you have a similar benefit just with neoadjuvant chemotherapy and pembrolizumab, and how much each of these parts of the treatment contributes. What we are delighted to see is obviously a benefit in the first instance.
So we have another question there. Yes.
Lynn Peterson with Trends in Medicine. So with multiple CDK4/6 available, does it really matter what's on your schedule or your formulary, as long as you're using a CDK4/6? Are they interchangeable in that sense? Is it a class effect? Yes, and understanding that we have data from one, but in practice, it may not be what's on your formulary, so do you think it really matters?
So I think the honest truth is we don't know yet. We haven't followed enough patients in enough trials long enough, and the cross-trial comparisons that we have currently available are certainly frail in terms of being able to draw interpretations from them. I think Dennis and I were talking about this actually before we came in. I think one thing that impresses us from a scientific standpoint is the consistency of the hazard ratios across trials. Whether you're looking at PFS endpoints or the emerging now OS endpoints, there's a remarkable consistency to the hazard ratios. So in terms of primary efficacy endpoints, I don't think that we have anything that would tell us that any drug is clearly superior to any other drug. There are clear differences in toxicities between the different agents that may favor one over another in a particular clinical situation. But I don't think we can yet say that one drug is superior to any of the others from an efficacy standpoint.
Professor Slemon, any comment?
Yeah, I think that I largely agree with what George has to say. I think one of the things we're learning about the class is it appears for breast cancer that inhibition of CDK4 may be more important than inhibition of CDK6. And in that context, ribociclib and abemaciclib, certainly in very clear preclinical studies, have that differential ratio, whereas palbociclib is basically equivalent in the inhibition of 4 and 6. So it gives you a bit of a different toxicity profile, but it also may give a bit of a different efficacy profile long term. But I absolutely agree with George that we don't have data yet to make a call like that.
So Nadia, would you like to comment on this?
Yeah, I think I couldn't agree more. And maybe just one point, looking at this in a pan-European way or even across the Atlantic, I think we're struggling with is that in a lot of countries, patients don't have access to either drug, and I think it would be preferable if they get access, and then we can sort out these differences in the meantime. So I think from a clinical point of view, what we do right now in the clinic is to sort of see how we like the management and train our staff to manage one or two of these drugs and then stay with them. But from an efficacy standpoint, we cannot say that one is superior to the other.
So another question in the back. Yes.
Aude Le Croupier, Medscape. Could you explain in more details what are the differences in toxicities for these drugs? For the
so George and Dennis, maybe you can summarize the different toxicities.
So all of the drugs have neutropenia as an issue. Abemaciclib tends to have less neutropenia than the other two. In contrast, abemaciclib tends to have more in the way of diarrhea than the other two. Ribociclib has a prolongation of the QTC interval, which is largely a paper toxicity in that it's something we measure on EKG and then manage appropriately. I would say there's emerging data, not quite strong or consistent enough yet for us to know the exact degree, but emerging data that both deep venous thrombosis and interstitial lung disease are rare events for all of these drugs, but are also present for all of these three drugs.
I think the report that came from especially in Japan and the population with abemaciclib with the pneumonitis has made the regulatory agencies particularly careful. Everyone has gone back and looked at their interstitial lung disease and pneumonitis data. And as George said, the incidences are very small, as we presented in the Mona Lisa trial. But this is something that the regulatory agencies have asked everybody to watch for and be careful about. One always has to weigh the benefits versus the toxicities, and the magnitude of the benefits we're seeing with these drugs, I think, are sufficient that it warrants moving forward with caution and looking closely at our patients and following them, but the benefits clearly outweigh the disadvantages of the drugs at this point.
So we have time for a final question. Yes, here, please.
Thanks. Jacob Please from Vantage again. Just a quick follow-up for Dr. Schmidt. How do you explain the seemingly large difference in responses in the control arm, depending on whether patients were PD-L1 positive or negative? That strikes me as counterintuitive. Thank you.
So that's well established from many chemotherapy trials that the more immune cells a tumor has, the better respond to chemotherapy. And there's even a theory that some of the effects of chemotherapy may be driven by the immune system. So that's the numerous studies, for example, from the German group that have established a clear link between the amount of TILs, so immune cells in tumor, and the path CR rates. Now, patients who are PD-L1 positive, most of the PD-L1 staining is on immune cells in triple-negative breast cancer. So if you're PD-L1 positive, you're more likely to have immune cells in the tumor than if you're PD-L1 negative.
So, in conclusion, finally, first, I would like to remember that all the data